Good evening, guys. This is Dr. Sticky Notes. The well compiled handwritten notes will be given here. And guys, if you are new to the channel, please click on Dr. Sticky Notes and get subscribed to the channel so you can access numerous videos which are less than 5 minutes. At least see one video per day. I have also created playlist for each and every subjects, so get benefited. All the best, guys. Welcome back again. In this video we're going to see the different form of microscope and the differentiating point between them and the important one-liners which has been recently asked many times in FMGE, NEET, PG, INICET, etc. I hope you will be well versed in this topic, so kindly take notes along with me. Yes, we can start. Let's start with the source. Yes, source is transmitted light. Whereas in dark field microscopy or dark ground microscopy, the source is reflected light. What is the source in fluorescent microscopy? UV lamps. Now LEDs are used. Okay. What can be the source in electron microscopy? The name itself implies electron beams. And the next we're going to see the types in each microscope. Yes, light microscopes are of 3 types. Okay, first, compound microscope, and the next, phase contrast microscope, and the next, inverter microscope. Let's see the individual use of each microscope. Of course, compound microscope is most commonly used microscope which we see in medical colleges in our second year, which has been used for gram staining and acid fast. Okay, phase contrast microscope is used for seeing the internal structure. Inverted microscope, it is exclusively used for viral cell lines. They are just viral infected cells. To see that, inverted microscope can be used. And the next, dark field microscope. Dark field microscope has no types. Why it is called dark field microscope? Dark field microscope is called dark because it has dark background. Okay, what is the use of dark field microscope? It is used to see a tiny locomotive structures or very thin structures. Yes, like flagella, are used to see organisms like spirochetes. Next, we are going to see about fluorescent microscope. In fluorescent microscope, it is of 2 types. One is focal microscope, other is confocal microscope. This focal microscope plays an important role in seeing the fluorescent stained structures. This confocal microscope plays an important role in seeing 3D structures. Very important question. And the next we're going to see electron microscopy. Electron microscope uses vacuum, so No living organism can be placed inside, only dead organism can be placed inside, keep that in mind. And what can be the types? Transmission electron microscope, scanning electron microscope. This transmission electron microscope plays an important role in seeing the internal structures. Scanning electron microscope is used to study the 3D structures. The original image will be black and white only. Then you can ask one doubt. In coronavirus picture, the electron microscopic picture was very colorful. Yes, you can make it color, but actual image is black and white. You can make it color with the help of software. So that's the point here. Okay, finally, as a last point, we can see the resolution for each microscopy. First, light microscopy. The resolution is 0.2 mu meter. What is the resolution for dark field? The same, 0.2 mu meter. For fluorescent, same, 0.2 mu meter. Okay, then for electron microscopy, here comes the difference. Both are seen in nanometer resolution. So that's why it is called electron microscopy. Okay, the transmission electron microscopy is of 0.2 nanometer resolution. Scanning electron microscope is of 20 nanometer resolution. Important MCQ are the resolutions. That's the topic ends today. This is the first microbiology video, I think. If you are taking a separate note for our YouTube channel, it's well and good. Then if not, please write in a paper, paste it on your wall or study table. Okay guys, thank you. All the best. Please take care of your parents at home. Thank you.